actually what I'm going to talk about today are two most recent papers that we published in our group. And so therefore the title is a little bit more generic. So it is more kind of related with investigating human fetal blood development at the single cell level. And so, so the overarching question in my group is how do blood cells develop, and how is their development and their function determined or influenced by their environment? And this is a very important question, both in the context of regenerative medicine, because at the moment we don't have efficient ways to expand stem cells for their use in transplantations, but also in terms of the pathologies and understanding what goes wrong, for example, in the case of blood malignancies in the context of the differentiation. And so to address these questions, as mentioned in my group, we are using zebrafish as a model system, and more recently primary human tissues. And so in that sense, we are using tumor samples, but also fetal samples to address the question of human fetal hematopoiesis. And what I'm going to talk about today are two studies that we performed in the last couple of years, and they have been published this year, where we looked at the human fetal hematopoiesis using methodologies that we normally use in my lab. And this is single cell RNA sequencing, single cell ATAC-Seq, single cell in vitro differentiation assays, but also whole genome sequencing more recently. And so just to kind of give you a brief overview of the what is known at the moment about the human fetal blood development. So the first hematopoietic stem cells are specified at 27 days post-conception in the area of dorsal aorta. And then these stem cells and progenitor cells will move into the fetal liver at around 4 weeks post-conception. And then in the fetal liver, the stem cells will expand in their number. They will self-renew and increase in their number, but they will also differentiate to give rise to all mature cell types, which are important to support the growth of the fetus. And then around 10.5 weeks, these stem cells and progenitor cells are going to move into the bone marrow. And then after birth, the bone marrow will be the only site of hematopoiesis in adults. However, there is an interesting period up until birth when the fetal liver and bone marrow are actually both like working in terms of the production of the blood cells, which gives us a unique opportunity to look at the two different hematopoietic sites and kind of what is similar or what is different in terms of the hematopoiesis in these two sites. And so to address these questions, the way we designed our experiments was to collect fetal liver as well as hip and femur, so to look at the bone marrow from human fetuses that were acquired through abortions. These were healthy fetuses from 17 to 20 weeks post-conception. And then from these different hematopoietic organs, we were sorting hematopoietic stem and progenitor cells as well as mature blood cells using either very broad markers for committed and non-committed progenitors such as CD34 and CD38, or we were using really well phenotypically defined blood populations based on the literature. And so altogether we had around a panel of around 23 different sorting strategies that we applied across 15 fetuses and these various tissues. And then we performed single cell RNA sequencing, single cell ATAC-Seq, as well as single cell in vitro differentiation assays to see the lineage output of these cells. And so to cut the long story short, what you can see here is one of the inferred differentiation trajectories where each of these dots represent a single cell. And so this is from single cell RNA sequencing experiment. And obviously these different colors represent different cell types that we were able to identify in our data set. And so what came clear from this data set was that at the tip of this trajectory, as expected, we were finding hematopoietic stem cells that were including a highly proliferative population of stem cells. But then downstream of these hematopoietic stem cells, we were seeing 3 oligopotent, highly proliferative cell populations that we defined as MEMP. So these would be progenitors that give rise to megakaryocyte erythroid cells and mast cells, so MEMP path. Then granulocyte path, so those would be progenitors give rise to all the granulocytes, and then LMP path, so these would be lymphoid myeloid cells that gave rise to lymphoid cells, but also there are monocytes and dendritic cells in our population. And so when we looked then at the, like you can see in this heat map, when we looked across the pseudotime, across all these 3 different paths, some of the marker genes that we identify in our data set, what we could actually see is that indeed populational cells that we defined as stem cells had a high expression, as we would expect, of CD34 cell, PROM1, and genes like MLLT3. And then these are being kind of slowly downregulated as these cells are differentiating. Whereas, for example, in the case of MEMP path, as these cells start to go into MEMP progenitor cells, they start to express KLF1, transferrin receptor. And then as they, for example, further differentiate into megakaryocytic lineage, we would see high expression of genes like ITGA2B or CD41. So all that suggested that, at least by looking at the expression of the marker genes across pseudotime, the inferred differentiation trajectory in a way made sense. What was actually interesting when we looked at the data set was also that in the population of cells that we defined as stem cells, we were not seeing expression of low level of expression of a lineage specific genes, which has been previously reported and defined as lineage priming. So basically what we were seeing is that this kind of lineage specific genes, which are important for the functional properties of various lineages, let's say megakaryocytes or erythroid cells, actually started to be upregulated only in the progenitor compartment rather in the stem cell and multipotent progenitor cells, suggesting that potentially this transcriptional priming that was previously described is not really happening during fetal development. Now, all the all the annotations that I was showing you were obviously done based on the computational analysis, and we were actually very keen to try and do some sort of functional validation, and in particular to focus on the population of stem cells and try to define novel markers. So as I said at the beginning of my talk, what we did in this experiment is that we were sorting phenotypically well-defined various blood populations, including stem cells and progenitor cells, and our transcriptional analysis was actually showing that these populations of cells were actually very heterogeneous. So there was a clear need to define a new panel to refine the sorting strategy for stem cells during fetal development. And now looking at our single cell RNA-seq data, we were able to define actually a new panel that we thought is better defining stem cells. And this is what I'm calling here CDREF. And so what we did is we defined, based on single cell RNA-seq data, a new panel for sorting stem cells, and then in the next few samples we were sorting these. We were sorting these CDREF cells, and then part of these cells we used to do single cell RNA sequencing, and the other part of the samples we were sorting as individual cells on either MS5 feeder layer, which is a mouse bone marrow feeder layer, or on mesenchymal stem cells that we isolated from these fetuses from femur. And then we were growing these individual cells for 15 days. And then after 2 weeks, we were looking at the lineage output. And so the first thing that came out of the single cell RNA sequencing is that when we looked at these cells and plotted them back on our UMAP that I was showing you earlier, 90% of these cells fell back into the stem cell compartment, suggesting that really CDREF cells represent transcriptionally highly homogeneous population of cells that represent stem cells. But more importantly, when we looked at the lineage output, a large portion of these cells were giving a multilineage output, suggesting that indeed what we are capturing are probably stem and progenitor cells. And so next we wanted to actually see if there are any differences in the functional properties of the stem cells defined by this CDREF panel in the fetal liver and the femur from the same fetus. So we were using the fetal liver and the femur from the same fetus, meaning they're being matched in terms of the development, but also individual fetuses. And so as expected, what we were seeing is that when we looked at the CD34 positive, CD38 positive cells, which would be committed progenitor cells, there were more cycling than a non-committed CD34 positive, CD38 negative. But what was kind of more interesting is that when we looked at our CDREF stem cells, when we were isolating them from femur, a smaller proportion of cells were cycling compared to this same cell population from the liver by looking at an MKI67 staining. in a way suggesting that as the stem cells are migrating from the fetal liver to the femur, they're slowing down in their cycling already. And so these experiments were done again on the fetuses between 17 and 20 weeks post-conception. Now, as I was mentioning in one of the earlier slides, we didn't really see much difference in terms of we didn't really see much of what we would call transcriptional priming in our stem cell compartment, and based on a transcriptome. And we wanted to see if there are any differences or heterogeneity that we could capture based on in terms of the chromatin accessibility and epigenetics. And so to do that, we decided to do single cell ATAC-Seq, which is single cell chromatin accessibility analysis on CD34 positive, CD38 negative populations. So looking at the non-committed progenitors collected from the fetal liver and from the bone marrow from 3 different fetuses, again, between 17 and 20 weeks post-conception. And so in this case, by using a shared nearest neighborhood modularity optimization-based clustering, we were able to identify 7 clusters that are here colored in different colors. And they were actually distinct in terms of differential accessibility peaks that they had. And obviously the first challenge here was to relate these different clusters that we are seeing in our single cell ATAC-Seq data to the cell types or annotations that we were seeing in the single cell RNA-Seq data. So to what cell types, so to say, these different clusters correspond to. And the first thing, and this actually turned out to be very challenging. So the first thing that we tried to do was to look at the marker genes that were very good at explaining different populations of blood cells in RNA-Seq data. So for example, CD34 or CD52 or cell PROM1, all these genes that we were seeing to be highly specific for the HSC population in RNA-Seq data. And unfortunately, that didn't really that was not very helpful, but because when we looked at the accessibility of, chromatin accessibility of these genes, there was not much that could actually tell us what is the difference between these different clusters. Actually, we were seeing more global patterns. So for example, that overall, there was a higher accessibility of these genes in cluster 1. But then, for example, when we looked in cluster 3, the accessibility was lower, and furthermore in cluster 5. And then, for example, cluster 4 was somewhere in between. And so really looking just at the marker genes, which are normally very good at explaining or very helpful in annotating cell types in RNA-Seq data, were not actually that much telling in the single cell ATAC-Seq data. So we decided actually to have a different approach, in which we decided to look again at the differentiation trajectories. So similarly, like in the RNA-Seq data, we tried to plot cells based on the changes in the chromatin accessibility that is happening during differentiation. And this kind of resulted in seeing 3 main paths, which I'm going to just kind of explain here in terms of the numeration, because at this point it was still not clear what these are. So we had the path which was defined by clusters 1, 2, and 3, and then second path which went with clusters 1, 4, 5, and 6, and then the third path, which was 1 and 6. And so when we looked now at the overall chromatin accessibility in each of these clusters, we were seeing that there was the highest accessibility in cluster 1. And then as we were moving along these different branches, the overall chromatin accessibility went down. And this is the first time that we started actually thinking that possibly cluster 1 were our stem cells, because it has been previously shown that in pluripotent stem cells, there is a higher chromatin accessibility, which then actually goes down when the cells start to differentiate. And so we, in a way, kind of assumed or postulated that actually maybe similar mechanisms are happening in the stem cells. So now, to explore this further, we decided actually to use a ChromeVar to actually look at the most variably accessible transcription factor motif sequences in our 7 clusters, and then to plot these across these pseudotime data or differentiation trajectory that I've just mentioned. And so we looked what is happening with this with the transcription factor activity, how it changes when we move from cluster 1 to from cluster 1 to 2 and 3, or from clusters 1 to 6, or 1, 4, 5, and 6. And this is something that was actually very insightful, because we've noticed that in cluster 1, which we defined as most probable stem cells, we were seeing high transcription factor activity for GATA1, TAL1, and KLF1. But then as the cells started to move to cluster 2 and 3, we were seeing high transcription factor activity for the genes which were important, transcription factors which were important in myeloid and lymphoid lineages like HDF4, ID4, or IRF8. However, when we looked, for example, in clusters 1 and 6, we could see that there was a clear increase in the transcription factor activity of transcription factors like GATA1, TAL1, which are important for the megakaryocyte, erythroid, and mast lineage. And similar was for clusters 1, 4, 5, and 6. And in that sense, this kind of suggested that potentially, whereas our cluster 1, our stem cells, cluster 6 probably represents our megakaryocyte, erythroid, mast cells, where the clusters 2 and 3 are more linked with the myeloid and lymphoid lineages. Now, to further examine this, we decided to use another tool to integrate single-cell RNA-Seq and single-cell ATAC-Seq data, which is based on, it was at that time kind of recently published paper where they were using anchors between two different data sets, like identifying anchors between two different data sets, in this case, single-cell RNA-Seq and single-cell ATAC-Seq, to find pairwise correspondences and then transfer the labels from one data set to another data set. And so we use our RNA-Seq data set to actually transfer the labels and identify different populations in single-cell ATAC-Seq data. And so when we did that and then plotted the frequencies of these different cell types in our data set, what we were seeing is a very similar frequency of different cell types that we were seeing in the RNA-Seq data, suggesting that most probably this transfer of labels was correct. And so basically around 50%, 57% of our cells were stem cells and 18% were highly proliferative stem cells and so on. Now, the surprise came when we then plotted these, when we looked at the distribution of these different cell types across 7 clusters. What we've noticed actually is that the stem cells were distributed across all 7 clusters, and the only place where we could see a high enrichment in a particular population of cells was cluster 6, and they were highly enriched for MEMPs. So this was good in a sense that this is what we were already predicting based on our previous analysis, but kind of suggested that actually on the population of the stem cells, there is a lot of heterogeneity. And when we then looked at some of the transcription factors, which are important for different myeloid, lymphoid, or erythroid lineages, and we looked at their transcription factor activity across different these 7 clusters, we could see that there was a lot of statistically significant variability, suggesting that in the population of the stem cells that we could see is homogeneous on a transcriptional level, there was a lot of different subpopulations that differed in terms of their chromatin accessibility, in particular transcription factor activity for the lineage, for different relevant lineages during differentiation, suggesting that this cell fate choice is maybe happening not on a transcriptional level as previously suggested, but on the epigenetic level. Now, to explore this further, we then actually wanted to see how the expression of how the expression of the various genes that are in this particular case in this heat map, we were looking at the GATA1 regulant genes. So these were the genes that were regulated by GATA1 transcription factor. So how the expression of these genes changes as the cell are differentiating from stem cells to MEMPs, and then, for example, in this case, megakaryocytes, and what is actually happening also in terms of the chromatin accessibility at the promoter level or enhancer accessibility, which we defined here, plus minus 50 kb from the transcriptional start site. And what was obvious is that in cluster 1, which we defined as our stem cell cluster, we could see that there was already high promoter accessibility for all of these GATA1 or majority of these GATA1 target genes, whereas on the RNA level, we couldn't see the expression of these genes. And so in a way, this all kind of, again, supported the idea that the first changes that are happening in the stem cells or heterogeneity in the population of stem cells is actually first visible on the level of the chromatin rather on the transcriptional level. And so I'm going to finish this first part of the talk with just some kind of key points that came out of the study, where we were actually able for the first time to generate epigenetic and transcriptional landscape of human fetal bone marrow, but also fetal liver cells, because in previous studies, they were not looking at the chromatin accessibility of these cells. And what we were able to show is that the stem cells and progenitor cells actually differentiate into 3 distinct oligopotent progenitor populations, which are highly proliferative, and that changes in the motif accessibility or transcription factor activity, how we define it in stem cells, precede activation of lineage-specific transcriptional programs and their cell fate choices. I didn't really mention that, or I didn't point out during the talk. We've noticed 2 opposing patterns of motif accessibility for 2 different TAL1 binding sites that seems to be active in erythroid versus myeloid and lymphoid lineages. And finally, based on our single-cell RNA-Seq data and single-cell in vitro differentiation assays, we were able to actually refine the sorting strategy to isolate human fetal hematopoietic stem cells with around 90% purity on a transcriptional level. I should point out that this panel is really something that is highly reproducible and does really sort stem cells, but at the particular stage of development, meaning from 17 to 20 weeks post-conception, because some of the studies that we are now performing are actually clearly showing that these cell surface markers are very dynamic and changing rapidly during the early stages of development. So I'm not going to actually completely change the topic. I'm going to stay still I'm still going to kind of talk about human fetal blood development, but really from the developmental point of view, rather in terms of the differentiation processes and transcriptional changes. And so what we were actually also interested in looking at is what is happening from the point of development, where we know actually that many childhood leukemias have their origin during fetal development. And what we actually don't know is what are the patterns of the mutation acquisition during normal fetal development, to really understand what is happening in the context of blood malignancies. And on our way to kind of do this and address these questions, we actually had some also other interesting discoveries. Now, the whole study was actually based on a very simple concept, and that is that somatic mutations actually accumulate in cells' genome over successive cell divisions. And so any of the progenitor cell which divides and acquires new mutation, that new mutation will be inherited by all the progeny of these cells. And so what that practically means is that if we collect blood at any point during our life, whether that's fetal development or later on during adulthood, and we know all the mutations that are present in these individual cells, we can actually reconstruct a phylogenetic tree. And so this is exactly what we did in our study. One thing to actually bear in mind is that when one is reconstructing this phylogenetic tree, you're actually going back to the very, sorry about this. We are going actually to the very beginning of the development, meaning that these mutations start to be acquired from the first division of the zygote into 2 blastomeres. And so I'm going to just give you a brief overview, very kind of superficial, very brief overview of a human embryonic and fetal development, which is relevant for understanding the experimental design that I'm going to explain in a few minutes. So basically, as I said, the zygote is first dividing into 2 blastomeres, and then these 2 blastomeres will divide further. And then around 100 cell stage, there will be a first event, a crucial event, where we can physically see separation of cells, which will give rise to trophoblast, and cells which give rise to inner cell mass. And the cells which will give rise to trophoblast are destined to give rise to placenta, to the fetal part of the placenta, but also to chorionic sac, whereas inner cell mass is destined to later on give rise to the embryo proper. And this is very important because all the mutations that are acquired in the cells of a trophoblast will never actually, after this separation happened from the inner cell mass, will never be present in the embryo, and the vice versa. So all the mutations that are are acquired after separation of trophoblast and inner cell mass will only be present in the embryo and never in the tissues which have a trophoblast origin. And so the inner cell mass will later on give rise to hypoblast and epiblast. It is known that hypoblast gives rise to yolk sac, but it has been hypothesized, and it's something that has been debated for the last 5 decades, whether hypoblast gives rise to extraembryonic mesoderm in human, which is actually giving rise to mesenchymal core of placenta villi and primitive blood. So this is the first blood that is formed in the fetus and is independent from the stem cells that I was explaining in the earlier talk. And then there is also epiblast, which will give rise to ectoderm, mesoderm, and endoderm during gastrulation. Now, we know already a lot about the human embryonic and fetal development, mostly from the careful microscopic observations at early stages of development or in vitro studies more recently up to the point of gastrulation. However, what kind of our study gave us, in addition to these traditional ways of looking at the human development, is the fact that when these somatic mutations are acquired in any particular cell, in any actually given cell, all the progeny of that cell will maintain these mutations. And it's some kind of stamp that allows us actually to later on during development see what particular kind of group of cells became later on during organogenesis. And so to kind of utilize this to its maximum, the way we designed our experiment was to actually take 2 fetuses, one which was 8 and 18, and the second one which was a little bit older, 18 weeks post-conception. And then we isolated hemopathic organs, liver and bone marrow if it was available. And then from these, we sorted individual, so single cell sorted hematopoietic stem cells and progenitor cells, and then we grew them to the colonies. And then we did a whole genome sequencing and somatic mutations calling. And based on these somatic mutations, we reconstructed the phylogenetic tree. And then, but at the same time, from the same fetuses, so these are now matched organs, we actually collected organs which have a different embryonic origin. So for example, skin, or epidermis, which has ectodermal origin, or gut, which is endodermal, and so on. And we sectioned these tissues, and then we did a laser capture microdissection of areas of interest so that we know that the structures that we are looking at have a clear and well-known embryonic origin. And then we did a targeted sequencing to see if the mutations that we identified in our blood colonies are present in these various tissues. And then that information went back into understanding or interpreting our phylogenetic tree. So again, to cut the long story short, at the end, we sequenced around 500 whole genomes from these 2 fetuses. And what we could learn from this is that around at 8 weeks post-conception, we could already see that there are around 20, a little bit more mutations accumulated in stem cells and progenitor cells, whereas this number goes to 42 by 18 weeks post-conception. And secondly, as previously actually shown, we identified a higher than expected proportion of single nucleotide variants in the variants in unannotated regions of the genome compared to the protein coding regions. Now, we use these mutations to, as I said, reconstruct the phylogenetic tree. And so what you can see here is a phylogenetic tree for 8 and 18 weeks post-conception. And so just to briefly explain what we are looking here at. So on the y-axis, on the right-hand side, you can see the number of mutations. And the length of these branches is proportional to the number of accumulated mutations. And each of these terminal branches actually is the colony that we sequence. So as I said, there were around 500 colonies. And then what is important to kind of mention here is that the top of our phylogenetic tree is a zygote. And then here I kind of labeled these 2 first blastomeres that were generated. And you can see the same here for the 18 weeks post-conception fetus. And so one of the first things that we could actually see, and it's very striking here, was that the contribution of each blastomere to the blood compartment, or actually to the different fetal tissues, was not symmetric. So we were seeing that in, for example, in one fetus, one blastomere was contributing to around 30% of blood compartment, whereas in the second one contributed 70%, whereas in the other fetus it was 99% versus 1%. And later on in some other studies, the same observation was made, kind of suggesting that actually there is a lot of plasticity in terms of the contribution of each of the blastomeres to the embryo, to the embryonic tissues. The second thing that we were able to see is what is the rate of mutation acquisition. And so what that showed is actually that in the first 3 divisions, there is a much higher rate of mutation acquisition compared to the later on. And it's equivalent to around 2.4 mutations per cell division, whereas later on it drops to 0.9. And as you can see here, actually, after the 3rd generation, there is the profusion of these polytomies, which can be in the context of phylogenetic tree, be defined as the cell divisions in which there is no accumulation of or no acquiring of the mutations, which can actually explain this drop in the rate of mutation acquisition. And so we don't have really clear understanding of the mechanisms behind this different rate of the mutation acquisition, but we kind of hypothesize that because this drop from 2.4 to 0.9 actually coincides with the zygotic genome activation and the transcription of the activation of the DNA repair machinery, expression of the DNA repair machinery in the zygote, that can actually explain drop in the rate of mutation acquisition. Now, what we couldn't say is actually when in development these mutations have been acquired. And this is where these different tissues that we collected were very useful. So just kind of going back to the earlier slide that I was showing, is that what we could actually see is that as the zygote divides to blastomeres and then their separation to various tissues, what we can actually expect is that the rate, the level of mutation sharing between blood, which is, for example, of mesodermal origin, will be higher with the tissues that separated from the blood antecedents later during development, whereas the tissues that separated from blood antecedents earlier during development. So for example, the first one would be the trophoblast, are the ones that we have the lowest level of sharing with our with the mutations that we detected in our blood colonies. And so we actually now wanted to see for all these various tissues that we identified, that we collected and did the targeted sequencing, we wanted to see what was the level of the mutation sharing with the colonies on which we did the whole genome sequencing. And so the result of that is actually presented in this phylogenetic tree here. So this is again the same phylogenetic tree as I was showing you earlier. So this is from 8 weeks post-conception fetus. It's just that we cut it here at 22 mutations so that we can focus on the upper part of the phylogenetic tree. And so here, this allowed us actually to look at the timing of the divergence of embryonic and extra-embryonic lineages during development. And so these different colors are actually mutations that are shared between our blood colonies and these various tissues. And so, for example, in red are the mutations that are shared between the tissues which have trophoblast origin and our blood colonies. And the only mutations that can be shared between these tissues are those that happen before trophoblast separated from the inner cell mass. And that's why in this legend here, we are calling these mutations that are kind of colored in red as those that happen prior to inner cell mass commitment. And so the same goes for all these various colors. So basically we were looking when these mutations, which of these mutations were actually shared with the blood, and they can only be shared prior to separation of that tissue from the line of descent that gave later on blood. And so that actually gave us more insight into the origin of the primitive blood. So the other way of presenting this information would be also to look at the cell fraction that contains mutations that are shared with these blood antecedents at the different stages of cell division. So these are cell generations. So the one cell generation is at 2 cell stage, then 4, 8, and 16. So we are basically looking here at the up to till the 16 cell stage embryo development. And so what you can see here are these different lines, which are showing the tissues which are coming from trophoblast, then placenta, villi, mesenchyme, which we know are extra embryonic mesoderm, peripheral blood, and then post-gastrulation tissues, like all these tissues that are coming from the ectoderm, non-hemopatic mesoderm, as well as endoderm. And so what we could see is that 90% actually of trophoblast lineages were lost in the 3 by 4th generation, whereas only 25 and 50% were lost from placenta, villi, and peripheral blood, and only 10% from the post-gastrulation tissues. And that kind of suggests that actually peripheral blood and placenta, villi, mesenchyme tissues separated earlier from the blood antecedents compared to the post-gastrulation tissues, and in a way suggested that actually they come from the hypoblast rather than epiblast like other post-gastrulation tissues. So another thing that we wanted to look at is, and kind of interesting observation that we made was that one of the questions that is not very clear is what is the contribution of the epiblast to various tissues like ectoderm, non-hemopatic mesoderm, and endoderm. And so here we looked at the patterns of shared mutations across these different tissues, and we noticed that the patterns of shared mutations in ectodermal tissues is distinct from that which is observed in non-hemopatic mesodermal and endodermal tissues. And so here again, you can see the phylogenetic tree of 8 weeks post-conception fetuses. What you can see here on the right-hand side on the y-axis is actually generations. So this is the number, again, first generation is a 2-cell stage, 4, and so on. And then what you can see here are the mutations which are shared between non-hemopatic mesodermal tissues or endodermal tissues or ectodermal tissues with the mutations that we were seeing in our blood colonies. And so what we could see for the mesodermal and endodermal tissues is that we detected mutations from the majority of blood antecedents up to the 4th, 5th, or 6th generation. But the contribution of each blood antecedent to the sequence tissue was very small. So you can see that actually in these pie charts, the cell fraction, the fraction of cells of these various tissues that contains these mutations that we identify in these blood antecedents is very small. However, if you look at the ectodermal tissues, then we can actually see that the smaller number of blood antecedents is actually contributing to higher proportions. So in these pie charts, we can see that the cell fraction is higher to the different ectodermal tissues. And in a way that suggested that the smaller number of epiblast cells contributes to the ectoderm compared to the mesoderm and endoderm. And so some of the main conclusions from this part of the talk was that what we could actually see is that during early human development, there are tens of somatic mutations that are actually accumulated. And as I was saying, like by 8 weeks, we can see around 20 mutations already in stem cells and progenitor cells. And that there is unequal contribution of each blastomere to the blood compartment and most probably to other embryonic tissues. We can see that the mutation rate is not constant during the development, so that it was much higher during the first 3 divisions. So it's around 2.4 mutations per cell division and then drops to 0.9. We were able to estimate the number of blood antecedents at the different stages of embryonic development and show some evidence that ectoderm originates from a smaller set of blood antecedents compared to the endoderm and mesoderm. And also our data highly support the hypothesis actually that the primitive blood in human originates from the hypoblast, and also that extra embryonic mesoderm actually in human originates from the hypoblast. And this is very different from the work that has been done and shown in mouse, where actually extra embryonic mesoderm and primitive blood originates from the epiblast, actually from the primitive streak. So I'm going to finish here. So I would like to thank my lab members listed here. And then the first part of the study was done in collaboration with Judith Zug from the Heidelberg, from EMBL from Heidelberg. And the second part, where we looked at the mutation sharing and using the somatic mutations as a way to do the lineage tracing during fetal development is done in collaboration with Peter Campbell at the Sanger Institute. And this work is funded by my ERC starting grant. So I'm going to finish here now. And
Terrific.
I can
Well, thank you so much. And thanks for that fantastic talk about both of those amazing stories. I wanted to open it up to see if there's any questions from the audience. Please don't hesitate to either raise your hand or put questions in the chat if there are any. Maybe I'll just start, Ana, with an initial question that I had. This finding of a non-constant mutation rate in early development is incredibly fascinating, and I was wondering if you could just talk about what you think are some of the underlying mechanisms, and also in light of the recent work from the Sanger describing how mutations might occur outside of the cell cycle. Do you think that there might be phenomenon that are occurring that are inducing mutagenesis? And also, as we think about implications for cancers, what do you think? Do you think we can always think about a constant mutation rate, or do you think we have to?
Yes, so I think, so I must say that, compared to my previous work, this was a kind of new adventure that I took, like to go into kind of more genomics rather than transcriptomics. And actually, yeah, so in that sense, we found it actually very interesting that to see that there is a non-constant mutation rate. So I think I was kind of saying that we don't really have a kind of clear understanding why is this happening, and so we can only speculate. So this is just kind of really kind of speculating, and that is that this kind of third cell division. So this is now at 8-cell stage. This coincides actually with the maternal to zygotic transition in human development, so that there is a genome activation in the zygote, and so some kind of idea would be that because there is now this translational machinery and the DNA repair molecules are being now kind of active in the zygote, that kind of helps to bring the mutation rate down. This would maybe kind of make sense if one was thinking that during each of the division, there is a dilution of the proteins which are being inherited by the mother, which I'm not sure. I mean, dilution is happening, but whether it is such that it would have this effect is I'm not sure. I think there is also some evidence that overall chromatin is more open, and so maybe it's kind of more prone to the mutations. And going back to your question about the acquisition of mutation independent from the cell division, so that might play a role. So I can't say that we know the answer, but we kind of think that maybe this is in line in this kind of overall much higher chromatin kind of openness, and probably this may be maternal to zygotic transition.
Great, well, thank you. I see a question in the chat from Mohammed Najia. And I don't know if Mohammed wants to ask it, but maybe I'll just try to paraphrase, I think, what was asked, which is, Mohammed was wondering, have you performed limiting dilution analyses or other analyses to quantify functional HSCs and compare those to the molecularly defined HSCs that you have?
Yeah, we didn't do any transplantation experiments. I think it's mostly related with the fact that these samples are very hard to get. It took us like 2 years to collect these 15 samples. And then secondly, the number of cells is very low. So the older the fetuses, obviously one has more cells, but they're more difficult to get. It just has to do with the fact that if women are aborting, they're doing that during earlier pregnancy rather than later. And so we never had actually a sufficient number of cells to do these type of experiments, nor abundance of samples to actually do them. But yeah, I think maybe. Yeah.
I see a hand raised from Ho-Jin Lee. Ho-Jin, do you want to unmute?
Hi, Ana. Really powerhouse talk there. I tuned in for one story and got a bonus story that was just as amazing. So really, really fantastic. Thank you. So along similar lines as Vijay's question about mutational rate, I'm actually curious that your data, it might be able to give some insight into what areas of the genome, or particularly what parts of genes, are more prone to naturally acquiring somatic mutations. And so in those, there was a large fraction of the somatic mutations that were in genes or protein-related genes or protein coding-related genes. I was wondering whether most of those somatic mutations were found in regulatory regions, in the coding sequence, introns, exons, et cetera. Do you have any idea about that?
Yeah, we tried really hard to find something interesting there. And actually, I think mostly we were seeing that it's kind of in non-coding regions. It was not really kind of even in a regulatory region. So I think that actually in a way, and I think there is some kind of previous evidence that I think I now stopped sharing, but I had this kind of bar where we were kind of looking at these unannotated genome and then related to the protein coding genes and so on. And it was actually lower than expected rate. You know, we were seeing less than expected mutations in these regions. So it was almost kind of that there was a higher chance of them happening in the parts of the genome which will not be detrimental, let's say, to the development of the fetus.
Okay, interesting. Thank you.
Are there any additional questions at all? Well, if not, thank you again, Ana, for that fantastic talk, and I'll pass it over to Liang to finish up.
Okay.
Thank you so much. I don't have any other question. Really wonderful talk, and thank you everyone. So
okay,
we'll see you in this.
For inviting me.
Right.
Thank you.
Thank you.
Bye-bye.
Bye.